I'm Jane Gilbert, President and CEO of the ALS Association. At least I can say that for the next 2 months. As I stand before you, this is my 5th public policy conference, and it will be my last as President and CEO of the ALS Association. I can tell you that the past 5 years have been the most remarkable years of my life, and I have appreciated the many wonderful friends and people I have met through this job. In fact, it really isn't a job, it's a labor of love. So welcome to all of you. As I always do when I stand in front of this group, I'm amazed at the number of people who take of their time and their talent to come to DC and be a part of this. And of course, I so appreciate those of you who are living with ALS, as well as your family and circle of caregivers. I'm also always struck by the fact that as I look out across the audience and see faces who have become dear friends, faces who are casual acquaintances, but all of us are united in the same effort to find a treatment and a cure for ALS. The one thing that always bothers me as I stand in front of a group like this is the faces that I don't see. Those people who have died over the last year, who have succumbed to ALS, and that makes me always so sad. But I would like to take just a few minutes here, a few seconds in remembrance, if we could, for those people who are no longer here with us. Thank you so much. And in their memory and in honor of all of those of you who are here, again, I welcome you to this, our 11th annual Public Policy Conference. I'm turning to Steve going, well, anyway, it's 10th or 11th. Thanks for coming, and please know that you are making a tremendous difference in what we do for this disease. Your efforts have provided people with ALS unprecedented access to Social Security, Medicare, and in many instances, veterans' benefits. You've raised awareness of the disease on Capitol Hill. It is no longer just somebody knocking on the door of a congressman. Many of you have become friends. You have become personal faces of people who are on Capitol Hill who advocate right along with us for the disease of ALS. It's truly personal. Your efforts have advanced ALS funding for research to the tune of $850 million in government funding over the past 10 years. And still, there's much work to be accomplished. Telling your stories, making it personal, going to the Hill, indicating to our Congress, your Congress, remember they live in your districts, how important funding for this disease really is and how important their vote for the things that we're trying to advocate for will make a difference. So we need to increase funding, and we also need to advocate for policies that will help us find a treatment and a cure for ALS, and that's why we're here. 75 years ago, Lou Gehrig gave his famous speech on the 4th of July and said he was the luckiest guy alive. 75 years is a long time, and it's amazing that we have not had more advances in this disease in these past 75 years. That's the story we have to tell, and that's where you come in and what will make a tremendous difference. We have a wonderful 75th anniversary campaign that I'm sure you are seeing or will see. It is around a baseball theme, as is our public policy conference this year. I hope those of you who have the baseball cards enjoy them, distribute them, and make wise use of them. These conferences are also being streamed online, so for those of you who are in our virtual audience, welcome to you as well. I'm not used to this vining, I believe they called it, but I do welcome all of you as well, and we're delighted to have you as part of this. We will also have a virtual advocacy day. So while those of you in this room are on the Hill, those of you who are at home can also make a difference by flooding the congressional offices with your letters to reinforce the messages and expand our message of finding a treatment and a cure. There are 130 people in this room or who are attending this conference who are living with ALS. And there are another 600 and some who are in this room today. Over the next 3 days, we expect 1,000 people to be part of this. And we don't do it alone. This is a partnership effort. Many of you have heard me say this before. This is a team sport. It is not about the ALS Association. We also include MDA, ALS Hope Foundation, ALS of Nevada, ALS TDI, the gathering of all of us together makes this the single largest gathering of the ALS community in the world. And that's a remarkable feat. So thanks to all of you for making the effort. Steve and his crew, and by the way, I want to publicly acknowledge Steve Gibson and his staff for the wonderful conference they have put together. They have put together a remarkable program over the next 3 days. It may seem like 3 years by Saturday morning. The expo, which will be held on Friday afternoon, is new this year, and I think it will be something that will be exciting and interesting for all of you. It's going to be like a trade show. There will be booths set up where you can learn the latest things about research, talk with the scientists directly, interact with our care services team, learn about equipment and advances in technology, and there will be things like programs for familial ALS. And if you're so inclined, you can even stop and visit with the leadership of the ALS Association, and we'll answer questions and say hello. The expo will feature an advocate lounge where you can come and meet and greet, rest a bit, talk with one another. We hear continually, we wished we had more time during the conference to interact with one another. And so this lounge will enable that opportunity. There will be a number of our sponsors who have booths, and you can take advantage of asking them directly, getting information from them, looking at their products, visiting with them. We certainly want to thank our sponsors for this because, as you can well imagine, this is not an inexpensive conference to put together, but it's an important conference. And we thank Biogen, Biotechnology Industry Organization, Cytokinetics, New Motion, Permobile, Pfizer, the Pharmaceutical Research and Manufacturers of America, PhRMA, Synapse Biomedical, and on a personal note, Ellen Phillips, who is a member of our Board of Trustees and helps fund part of this conference personally. I encourage each of you to stop by the booths and to meet and greet the people who are in the exhibit. When the expo opens, you will receive an actual passport. Many of you who attended last year's conference are familiar with this. You will go from booth to booth and have it stamped. And at the end of the afternoon at 4:30, oh, here it is. This is what it looks like. So this is your little passport. Please go have it stamped. When it's completed, you'll put it in a little box, and then I have the honor at 4:30 of drawing a name out for the winner of an iPad. And you must be present to win. So please understand that your passport must be stamped, and you must be present to win in the expo area at 4:30 on Friday afternoon. And we certainly will look forward to sharing an iPad with someone in the audience. Before I turn the program over to Steve Gibson, our Chief of Public Policy, I want to recognize the ALS Association leadership who are joining us for this conference. Not all of them are in the room today, so you certainly don't need to stand, but you will see their faces over the next few days, and I want to be sure that you acknowledge the members of our Board of Trustees: Millie Arnold, Andrew Brophy, Chris Brusalis, Doug Butcher, Don Casey, Sue Gorman, Marty Herbert, Tom Kettler, John Crave, Lou Libby, Kim Ann Mink, Warren Nelson, Stuart Oberman, Andrea Pauls-Bachman, Ellen Phillips, Judy Pratt, John Roberts, Bill Sofel, and the chairman of our Board of Trustees, Bill Thowett. I also want to recognize the leadership of our Board of Representatives, which is a robust addition to the ALS Association in the last 3 years. It is chaired by Ken McGonigal, the vice chairman is Toby Kuharski, and the secretary is Kevin Hemingway. Again, I want to thank Steve and his team for the tremendous work they've done on this conference. I hope you enjoy it. I know you will find great educational value in it. And again, I want to extend my thanks for the tremendous work you've done over these years of advocating on Capitol Hill. So I want to also recognize, finally, the staff of the National Organization. And I am going to ask them to stand, and there's a reason for it. So, National Organization, please stand. And if nobody stands, I'm going to be really mad. They are standing, and some around the peripheral of the room. Thank you so much. I didn't actually have them stand so that you could applaud them, although that's very much appreciated. What I did have them stand for is that I want you to recognize these faces. And if you need anything during this public policy conference, please grab somebody, including me, and we'll see that your needs are met. So again, thanks to all of you. I welcome you to Washington. I thank you for all you do. And let's go get them. Steve?
Thanks, Jane, and thank you all for really getting here early. I know some of my West Coast friends had to come in a day early just to make sure they're in time for this session. This is such an important session because the federal government is the largest funder of ALS research. In fact, approximately $70 million per year is spent on ALS research. And a lot of that is due to the people in this room who've advocated over so many years. In fact, since the conference began, we have over $850 million. So it's a huge accomplishment. I want to thank you all for helping with us that. So as you know, there's much more work to get done, but today we're taking a little break for you to understand what our federal government is doing on two very important projects and programs. First, I wanted to kind of do some reminders what this session is and what it is not. It's a session for you really to learn more about our ALS Research Program at the Department of Defense and our registry at the CDC. And they'll both have some really great presentations, and you'll be able to ask questions about it and other things. But it's not a time for us to question why we select our priorities or how we do it. That's for the session after this. You know, in one case, we have a federal government employee and another scientist funded by the federal government, so they, as you know, cannot advocate. So I want to just clarify the questions about how we came with our ask is things you can ask myself and my colleagues next session. The second thing is that both these individuals will be talking about a research project. And that research project is underway. And so they might be constrained by certain parameters through their IRBs on what they can and can't say. So I just want to take that in regard when you're asking your question. Welcome also to the people online. We are glad you are joining us, for those of you who can't come to Washington. Before we get started, though, I want to introduce somebody. It's a program manager at our Department of Defense Research Program. Dr. McGuire, would you please stand? For those of you who've been coming, probably recognize Dr. Forsythe. Remember Dr. Forsythe? She is no longer with our program, and we have Dr. McGuire, so we wanted to give her a nice welcome, and she will be presenting, obviously, in future conferences. Our first presenter is Dr. Nick Maragakis, who is not too far from here, up at Johns Hopkins, and is a world-renowned ALS researcher. He has been one of the recipients of the grants at DoD, and we're excited for him to share with us what he's working on.
So thanks very much. Thank you to the ALS Association for allowing me to speak with you here today. I have had the opportunity to receive the funding from the ALS Research Program through the Department of Defense, but also, I think importantly, grants from the ALS Association, other private organizations, as well as the NIH. And it really is a combination of all of these efforts that has allowed this research to go forward. Number two, I can tell you about a year and a half ago, I had the opportunity to get in front of 11 or 12 senators who were directly involved in healthcare policy. Chance to meet with them one-on-one in their office. I can tell you that your presence today and your presence this week is going to be, I think, even more meaningful to those people when they see patients and caregivers who take care of those with ALS. So I thank you for being here today, and for those of you traveling long distances. Third is that I'm not going to, I'm going to show you everything I have. There's nothing that I have I need to keep under wraps. What I will tell you, and I think that's really in the spirit of ALS research, because this has been such a challenging disease, I think it's probably the most, I guess, unified community from the standpoint of basic science, from the standpoint of clinical research, and so I do both basic science and clinical research and take care of patients as well. It's a group that's very collaborative and very open. So you'll meet a number of my colleagues, I think, throughout the next few days as well. And so I think it is a very collaborative effort amongst a large number of individuals. So with that in mind, I'm going to tell you a little bit, a little high altitude view, and I'm going to be speaking about a couple of big themes. The first one is with regard to iPS cells. These are induced pluripotent stem cells. And this is a relatively new technology. This is a technology that was really developed in patients or in humans in 2008 by Professor Yamanaka. And it's unusual for someone to win a Nobel Prize until sometimes years or decades following a discovery. Dr. Yamanaka won it last year, essentially, for his work in developing iPS cells. So this is a field that is growing rapidly, and I'll stress that a little bit later. So how are we using iPS cells, induced pluripotent stem cells, to understand disease, to look for potential therapies, and then finally for potential transplantation biology? And then part two will actually talk to you directly about a strategy we're working on to do stem cell transplantations in patients with ALS that was funded by the Department of Defense and the ALS Research Program. Yeah, I'm hitting the wrong number. There we go. Okay. So I think this is an important slide. And actually, can I see that? I don't know how well this is going to. So I'll use this side maybe. So I use this slide in particular to show you a couple of things. The top left corner has a pie chart that includes all ALS patients, and you can see that about 10% of patients carry that familial form. And you may be aware that in the last 2.5 years, a new gene representing a significant amount of ALS was discovered, this gene called C9orf72. If you were to take all patients with familial ALS, the vast majority of those patients are going to have this newly identified gene. This is a gene called SOD1, which many of you may be familiar with. It also represents a significant population. But taken together, familial ALS probably only represents 10% of all ALS cases, yet the vast majority of research has been and continues to be around a very small subset of patients with ALS. For the rest of you with sporadic ALS that doesn't run in families, how are we going to approach treating the disease, and how are we going to approach studying the disease either in a dish or using animal models for ALS? And I think for the first time in history, we have the opportunity to do so through using these induced pluripotent stem cells. So the next theme really is an understanding that's really evolved over the last 10 or 20 years, and that is, although we think of ALS as a motor neuron disease, or we traditionally thought of it as a motor neuron disease, the more we understand about ALS, the more we realize that it doesn't just affect motor neurons, but in some cases can affect nerve cells that are involved in thinking. But just as importantly, and perhaps importantly for disease therapies, we also recognize that it's not just these motor neurons in the spinal cord that are having trouble, but that other cell types in the brain and spinal cord can also contribute to the disease. And that includes cells like these oligodendrocytes, these supporting cells called astrocytes, which I'll touch on in a little more detail, and even cells that line blood vessels. The muscle may also have a role in ALS and its pathogenesis. The key here is that all of these different cell types offer us new opportunities to target specific therapies to our patients. So as a clinician, what are some of the observations I have and others in my field have that we don't necessarily understand, but I think is really important as we think about the disease? As a general rule, why does weakness in ALS seem to have a distinct anatomical spread? For example, patients who have trouble with swallowing or speaking are more likely to have facial weakness before they might have, let's say, foot drop. Why do some ALS patients progress more slowly over years to decades, while others have a more rapidly progressing disease? Why do some patients present with what we call upper motor neuron findings, maybe stiffness or spasticity, and others with weakness and muscle atrophy? Why do some patients have bulbar onset ALS, that is trouble chewing, swallowing, and shortness of breath at such an early stage, while others have disease that primarily affects the arms or legs. And why are certain groups of nerve cells spared? That is, the eye movements are spared until very, very, very late in the disease. Bowel and bladder function is generally spared. So I think the message here is that ALS is really no longer thought of as just a neuromuscular disease, but really a neurodegenerative disease that puts it in the same categories as diseases like Alzheimer's disease, Parkinson's disease, and others. So how good have we been at modeling ALS? And I think the answer is that we can clearly do better. Almost every ALS clinical trial, whether it be drugs or stem cells, has really taken its first steps towards clinical trial using this ALS mouse model that was really designed around the familial SOD1 mutation. So this is a mouse that can be made to have ALS by introducing a familial form of the disease. And these animals get weakness in their back legs and then their forelegs. And this is what we use to try to test drugs or test therapies. And while it's a universal model, we think we'd like to be able to do better. I'm just up here pressing random buttons like that. I don't even know if I'm doing anything. So the question then is, are there opportunities to use human-induced pluripotent stem cell-derived neural cells? So neural cells can be motor neurons or astrocytes or oligodendrocytes to address what we call the heterogeneity of ALS, what makes patients unique. And I think we will approach a time when we can talk about patient-specific therapies, or what we call individualized medicine. And we know that this is the case for cancer. Oftentimes, cancer patients get tested for certain genes, and those therapies that are tried on individual cancer patients are dependent on what type of, let's say, genes they carry. So what is an iPS cell? iPS cells are really derived from patients, and this is unique because we don't necessarily anymore have to deal with embryonic stem cells or fetal stem cells. I'll talk a little bit about those in a minute, but now these cells can be derived from patients. In fact, we can take patient skin cells, these are called somatic cells, we can take them from skin or blood, grow these cells up in a dish, grow skin cells up in a dish, and essentially reprogram them to make them into stem cells. And as we reprogram them to make them into stem cells, we can make them into any type of cell in the body, not just nerve cells, but also liver, heart, and so forth. So these iPS cells can then be used for a variety of different therapies. We can use them to look at patient cell-specific therapy. We can use them in a dish to potentially screen for drugs. And we can use them to try to understand the disease itself, that is human disease modeling. So rather than having one mouse with one type of ALS, we envision a day where we can make 500 cell lines from 500 patients, all with different forms of ALS. So this is the Hopkins ALS iPS cell library. And I had the opportunity to talk to your group back in 2011. At that time, we had taken 60 samples of skin from patients, had not at that point made any iPS cells. I don't know how well this projects, but what I can tell you today is that we've now sampled at least 120 to 130 skin cells from patients, and we've made a total of 53 iPS cell lines. Now, that may not sound a lot. There are some limitations. One of those limitations is that it costs about $10,000 to make every cell line. So really through a grant through the federal government, we were able to make, at this point, 53 different cell lines from individual patients. I also want to stress one more thing. On the left side are patients with the SOD1 familial form of ALS, as well as some other familial forms of ALS that you might see. One is called TDP43, one is called C9orf72, this new one I just mentioned. But potentially just as interesting are patients maybe just much like you with sporadic ALS, patients who have slowly progressing disease over 5 years, patients with typically progressing disease, patients with dementia, patients with primary lateral sclerosis, another ALS variant, and so forth. So with these kinds of patients that we can derive a whole library of different cells. Once we take those cells, what do we do with them? So in my laboratory and other laboratories around the country, we're able to actually push them in different directions. We can either make them these astrocyte cells, which are supporting cells of the brain and spinal cord, and I'll tell you about those cells in a minute, or we can make them into motor neurons, and we can actually perform these studies in a dish. So by directing these cell types into two different, what we call lineages, we can study ALS astrocytes, ALS motor neurons, and carry these experiments out in a dish. By the way, I will tell you it takes about 30 days or about a month to make a motor neuron cell. It takes about 90 days to make an astrocyte cell. If you were to do that from an animal model, it's about a couple weeks to make a motor neuron and maybe 3 weeks to make an astrocyte. So human cells grow much more slowly. But the question is, if there's such what we call phenotypic heterogeneity, that is differences between ALS patients, are all the mechanisms of why motor neurons degenerate similar, or could there be different pathways in different patients? So this is a current study that we're investigating. So we have now the potential for looking, in my laboratory, we're interested in looking at what makes disease go faster or slower. And for the first time, we can now take patients with slowly progressing disease, patients with what we call typically progressing ALS, patients with rapidly progressing ALS, let's say less than a year and a half, and then of course patients who don't have ALS at all. And we can take iPS cells, grow them in a dish, make them into motor neurons as the case may be, and now start to compare different rates of disease progression. So this is not something we could do even 2 or 3 years ago. So this is an effort now that's going on in my laboratory. And to give you an example, this is kind of color-coded here. These are 3 patients in green who have slowly progressive ALS. 4 patients in kind of this lavender, which have typically progressing ALS, 2 patients who have rapidly progressing ALS. All those patients have sporadic disease. We also have some with familial ALS, 3 which carry an SOD1 mutation has a very slow progression, and 1 who carries an SOD1 mutation with a very fast progression. So essentially we believe in a dish to carry out a clinical trial, if you will, in a dish to test what drugs may be better or what drugs may not work as well. To give you an example of these slowly progressing patients, one of these patients had ALS for 22 years, another one 41, another one 9 years. In the red here, you can see 2 patients with rapidly progressing disease. One had ALS for 20 months and the other one for 18 months. And of course, at the very bottom are patients who have no ALS at all. That allows us to compare and contrast what happens in a dish. So I would describe this to you as essentially being able to perform in a relatively short period of time an ALS experiment, or really a clinical trial, if you will, directly in our laboratory. So what can we do with these cells? We are now actually able to make very specifically these cells called motor neurons. And this is a pretty savvy crowd, so I'm going to show you a little basic science here. In this second panel here, you see all these green cells. These green cells are actually all motor neurons. This is not really a technique that we could have developed even 2 or 3 years ago. So we can get a high purity or a high concentration of these motor neuron cells to use them for study. I told you before we were able to push these iPS cells in different directions. What I hope you can appreciate is when we did this culture, what we were able to do is eliminate any of the astrocyte cells here in green. There are just a couple of them. And we were able to eliminate any of the oligodendrocyte cells. Now we essentially have a pure population of motor neuron cells, the cells that are affected in ALS. I will tell you similarly, we can make lots of cells in panel G while we make no cells in panel F. So we can push these cells in different directions to ask different scientific questions. So because it only gave me 2.5 hours to speak this afternoon, I thought I'd just... So I've shown you work in a dish, but the next thing we want to know is how closely do in vitro, that is, in vitro means in a dish, how closely do these cells that we make in a dish recapitulate what goes on when we would put them into patients, for example. That's called in vivo biology. So the next thing my laboratory is working on is trying to understand when we transplant cells into the spinal cord, in this case we're transplanting human cells into an animal, in this case a rat spinal cord, what happens to those cells? The first question we wanted to look at was not with ALS cells, but just ask the most basic questions between 2 different cell types. Human embryonic stem cells, these HESC, embryonic stem cells, that were made about 15 years ago, and human-induced pluripotent stem cells, these 2 cell lines down here. So these were just normal cells. These didn't have ALS. So we wanted to say, well, is one cell type better than the other? How do they act? Do they act differently once you put them into animal models or eventually into patients? So that's the question we've been asking, and we were just able to address this most recently in a publication. But what we were able to do is transplant these cells very specifically up into what we call the cervical spinal cord of rodent models. And we're actually able to pick out the human cells, separate them out from the rodent cells using a very slick technique called nanostring technology. So this is what I will tell you. There's 3 different colors here: black, red, and green. And each of these kind of things that don't make any sense, these are very specific, in this case, astrocyte genes. We were interested in making astrocytes. We're interested in astrocytes to try to protect nerve cells from harm. What I hope you can see is that the black bar represents embryonic stem cells, and the red and green represent human iPS cells. And what I hope you can appreciate is they look very similar. So this is actually exciting for us because it tells us, what I'm hoping is that we can finally, that the time has arisen we can use iPS cells rather than human embryonic stem cells, potentially for understanding disease and eventually for therapy. So the message here is that these cells behaved very similarly when we put them into animal models. And what I hope to show you here is that when we put these into the spinal cord, we can put these directly into the spinal cords of rats. And in fact, these cells, through some very specific human markers, we can show that they do in fact become the cells that we're interested in. And in fact, encouragingly, these cells can last for, at least as far as we've checked, can last for months in these rodent animals. So they do become the cells that we're interested in this case. Now, slide with a lot of data on it, but what I want to show you is this, and again, a lot of this may look like nonsense. All these are individual astrocyte genes. Right? These are astrocyte proteins that are expressed. Do the cells become what we want them to become? There's 2 lines there, a yellow line and a yellow bar and a blue bar. And each of these are 3 different cell types: embryonic stem cells, iPS cells in these 2. All right? What I hope you can appreciate is in almost every one of these figures that the blue bars are going up. You don't see a lot of yellow bars, but what you see is a lot of blue bars. And what these blue bars tell us is that when we took those cells from a dish, we put them into animals, they started to look very much like human mature astrocytes. So these are not immature astrocytes or immature cells that were growing in a dish. They now, when we put them into rodents, potentially someday into patients, they really look like the cells that we're interested in. And that may not sound like a big deal, but it's a big deal in the field. When you put a cell in, do you get what you paid for? Okay. All right. So these were normal cells. The next question, and these are just very what we call preliminary data that have just come out of my lab in the last week or so. If we take these same cells from patients, in this case we did it from 3 different patients, what do those cells look like after we transplant them? What I hope to show you is that these cells, if you compare the green, the red, and the blue, these are 3 different what we call astrocyte markers. And in every one of the patients, we looked at a control patient, that is a normal patient. We looked at a patient with the C9orf72, that is a familial case of ALS, and also a familial case of ALS that carry the SOD1 mutation. The pattern, those astrocytes, once we put them into the spinal cord, looked very similar. So those data would suggest that maybe independent of what kinds of ALS patients have, those cells become, again, what you paid for. What was a little bit of a surprise to us is when we looked at this green bar here, this was a patient with this C9 mutation. The actual cell survival was quite low. So this might suggest to us, at least in our preliminary data, that all cells aren't created equal with regard to their survival. So this is something we need to think about as clinicians and as scientists. If you're a patient with that form of ALS, do you want your own cells back, or do you want a normal cell type? Okay. All right. So that's really a very broad view of how we're using iPS cells to both model disease and to think about cell transplantation. I'm going to tell you about a program that's a little more proximal to the clinic, and that is as we started to think about stem cell replacement for ALS. So I will tell you that this is a project that we started thinking about 14 years ago, when stem cells were really first discovered both by investigators at Johns Hopkins Hospital as well as University of Wisconsin. So we thought it made sense because this was a motor neuron disease to replace motor neurons. But then we started thinking, well, how do we put cells into the right regions of the spinal cord? Can we make them into motor neurons? Would those same motor neurons undergo the same cell death? Would they make connections with other nerve cell types? What about regrowing those axons? We know that each individual cell grows about 1 millimeter to 3 millimeters per day. So it takes about a month to grow 1 inch. All right. So when you're talking about nerve cells that can be a yard long, I'll take that water. That's going to take some time. Reconnect to muscle, and then how about transplant rejection? So we thought maybe easier to afford a neuroprotective effect to surviving motor neurons. It is patients' motor neurons that are there, to protect those motor neurons and to keep them connected, at least in that point here in 2014, rather than trying to remake motor neurons. So let's plan on keeping the ones that are there. So in collaboration with really investigators from around the country, we've isolated these cells called glial-restricted precursors. And these are essentially designer stem cells. When we first started thinking about stem cells, we thought, stem cells, a stem cell, we're going to put stem cells in, they're going to make all the things we're interested in. So I'd like to think we've come a long way in that respect. So we've got these glial-restricted precursors. And these are designer stem cells that can only become either astrocytes, these supporting cells of the brain and spinal cord, or in some cases what we call oligodendrocytes, the cells that wrap around nerve cell processes. So we can identify these cells from animals, and we can actually identify them from human fetal tissue as well. And through a collaboration with a company called Q Therapeutics, we have a government-funded grant to take this to the clinic. So as we started thinking, if we were to try to get the biggest bang for our buck for patients with ALS, where would we try to target our stem cell transplantation strategies? We know that most patients run into trouble with respiratory failure. At the same time, respiratory function is a very reliable clinical way of measuring clinical progression in our ALS patients. It's almost a standard that we use when we do clinical trials in ALS. Many of you maybe have had to breathe into a tube to see what the breathing function looks like. So if we were to target a region in ALS, get the biggest bang for our buck, what would that region be? And so we just thought it made most sense to look at the cervical spinal cord. That is the region that innervates, or the nerve cells come from to innervate the diaphragm. That is the muscle of respiration. It turns out not only is this important in patients, but we can also recapitulate this in our animal models of ALS as well. So we then started looking at this ALS rat, and this was a rat much like the mouse that develops paralysis of the hind limbs and actually develops breathing problems as well. So what we did is we transplanted these cells. In this case, the first data I'm going to show you are rodent cells. These are animal progenitor cells going right back into a rat. We're able to target them very specifically into the spinal cord or into the neck region of these animals. And we were able to transplant about 300,000 of these cells at individual locations. And a number of years ago, now we were able to show that, in fact, we were able to get a big effect with regard to slowing the disease progression in these animal models. And it wasn't because we were replacing motor nerve cells. We were actually able to protect the ones that were already there. So we were able to preserve forelimb function in the rats. We were able to preserve breathing function, and we were able to push the disease course out. So using the same idea in rodent cells, we're now at the point at which we're saying, can we use human cells and can we translate this to the clinic? So we've actually done some initial studies using human glial-restricted precursor cells, put those cells into some of these same animal models and show that they have very much the same properties as the rodent cells, okay? So if you're thinking about designing a drug, you don't make a rodent form of the drug and a human form of the drug, you just make the drug. But when you're dealing with stem cells, which are very unique, you may have rodent stem cells, and you have to prove that they're the same or very similar to human stem cells, right? So with that in mind, we've approached the FDA and have been through the FDA, that's the Food and Drug Administration pipeline, to try to get these cells approved for patient use. And I will point out that there are two other large groups here in the States that are doing very similar strategies. My colleagues at Emory and University of Michigan, a company called NeuralStem, and then a group at Cedars-Sinai Hospital in California. What I'll tell you is all three of these groups have collaborated pretty extensively. Our delivery of these cells is all similar, if not identical. Each of the cell types we're using is slightly different, but I think it helps to say that it's been a very collaborative effort. What one has learned, we've shared with the others. So what keeps the Food and Drug Administration awake at night? Do human GRPs, glial-restricted progenitors, have similar properties to rodent GRPs, right? Are you dealing with, do the drugs look the same, or do they look completely different? Okay, so that's number one. Do human glial-restricted precursors form tumors? So we're transplanting these cells directly in the spinal cord. What we don't want are tumors of the spinal cord. Does the transplantation of human glial-restricted precursors result in what we call ectopic engraftment? If I put these cells into the spinal cord of my patients, I don't want these astrocyte nerve cells to end up in the liver, right? So that's unacceptable. And finally, is the surgical transplantation strategy for these cells safe? When you're talking about putting a needle in someone's spinal cord, that's a pretty big deal. All right, so we have approached the FDA, and I'll tell you, quite frankly, where we are right now. We are currently demonstrating that these cells are safe and they don't form tumors. This isn't something that happens in the Maragakis lab on the weekends. To give you an example, we've now contracted with another group called MPI Research. They transplanted 350 rats with these cells, and we monitor these rats at different time points. And it's essentially much like we would conduct a clinical trial. So this is the cells cannot be manipulated. I can't take them home or keep them in my pocket for a week. They have to be frozen, thawed, and transplanted, exactly how you would do it for a human clinical trial. Right? Part two is that we have to prove that the device is safe. And so in collaboration with my colleagues at Emory University, John Glass, and our neurosurgeon, Nick Boulis, they were actually injecting, actually John Glass wasn't, Nick Boulis, the neurosurgeon, was, but actually injecting what we call mini pigs. So why mini pigs? Because a mini pig's spinal cord approximates that of a human spinal cord. The FDA just wants us to show that, in fact, doing this procedure itself is safe. And that will just be a 1-month study. So I am happy to say that I think things are progressing in a, not fast enough ever, but I think we've met all the appropriate milestones, and we would very much like to see this to get to clinical trial. All right, so this does not happen in a vacuum. While the work I've shown you today is really a tenth of what's going on in my lab, this really happens through a variety of collaborators who have helped collaborate with stem cell studies. Mahendra Rao is the former chief of the stem cell section at the NIH, folks at Ohio State, Philadelphia, and Emory. We've worked with Q Therapeutics. Just for disclosure, I'm not funded by Q Therapeutics. I share a grant with them through the NIH. iPS cell studies are really collaboration with a number of different investigators really around the country. And of course, none of this could be done without the members of our ALS clinic at Hopkins and really patients like you. I think it's, in addition to clinical trials, it's an opportunity when we get iPS cells from our patients to once again make that connection between closing the gap between basic science and patient care. And then finally, we have the Ansari Center for ALS Research at Johns Hopkins, where we essentially generate these cells and distribute them really worldwide. And of course, this couldn't happen without our funding from the Department of Defense and the ALS Research Program, the ALS Association, and a number of other organizations to see this through. So I think I'll stop there. Thanks.
Great. Well, thank you. Any questions from the audience? Yes, Ted?
The injections, they're making you do more in pigs, you said? You having to do more injections in the pigs, you said?
All right, so why do we have to go back and do injections that have already been performed?
Yeah.
Yeah. So it turns out that, it's a good question. We asked the same question. So it turns out that because it's a different product, essentially, because it's a different cell type, how could those behave differently? And so the answer is because it's a brand new drug, if you will, even though the delivery device is the same, it's a brand new drug and has to be studied separately.
Okay. Thank you.
So, right. So the question is this strategy the same as the Emory, Michigan group? And the answer is yes, and it's really a collaboration. We're hoping that when we launch our trial at Hopkins, that the Emory actually will be a part of that clinical trial. So these are all shared efforts. The strategies are very similar, not by accident, quite frankly. It helps ease the getting through the FDA. And quite frankly, our cells we've not demonstrated are very effective in getting into the spinal cord if injected just into the spinal fluid. So at this point here in 2014, I think it's got to be right into the spinal cord or brain, as the case may be.
Is this procedure similar to the one the FDA just approved last week with Brainstorm's Neurone Technologies? And would the questions that you seemed to ask of the FDA, or the FDA is asking of you, seemed similar that they asked of them, and could that speed it up at all?
So their strategy is different in a variety. Their strategy is much more different than the other three that I mentioned, quite frankly. So they used a very different stem cell type called the mesenchymal stem cell, which is derived from bone marrow of patients rather than nerve tissue. So that's number one, and that's a big difference. Doesn't mean it can or can't be effective. It's just a huge difference between those cell types. The neural stem group and the Cedars-Sinai group and the Hopkins group are all using nerve tissue. The second one is that they're delivering it into the spinal fluid itself through a spinal tap, much like a spinal tap or a lumbar puncture. Those are two kind of fundamental differences between the two studies amongst other smaller ones. The studies, I don't know what the FDA asked them, so when you're dealing with a company, actually it's all kept, it doesn't have to be revealed. And the FDA doesn't have to tell me what they told somebody else either, right? So I think that those are two big things that I would say are different. Different cells, different delivery.
Question?
Thank you. Will your study be applicable to bulbar palsy?
So the question is, will our strategy be applicable, I guess, deliverable to patients with bulbar onset ALS? I think that's the question. So we haven't necessarily defined the nuts and bolts. My anticipation is because of where we're putting the cells that we won't necessarily restrict for different forms of ALS. The number one concern with any clinical trial is safety, and that's highest on the list, is to make sure that we don't make certainly patients worse. But my hope is that we'll do both bulbar and spinal onset ALS.
The IPS lines that you're developing at Hopkins, is with the cost so high, is there a strategy you're going after as far as like what types of patients, particularly in the sporadic ALS, that you'd like to go after? And then what's the next step for those? Maybe biomarker studies or something like that? Because I bet anybody in here would be willing to give you a bit of skin for the...
Right. No, I think so. You're absolutely right. We've been doing both. We started off taking every patient we could get in the clinic. We've now, just for practical purposes, have gone to patients with, let's say, unusual forms, slow or fast. Those have been two of the things we've been interested in, primary lateral sclerosis versus lower motor neuron only. But we've taken a large number of sporadic patients. What I will tell you is I think the time is soon coming where we won't have to give skin anymore, but we'll actually just get them from blood. And many of you, I think maybe some of you may be giving blood this week. So my anticipation is that we'll be able to do this from blood as well. I would certainly welcome anyone who's interested in coming. We'd be certainly happy to take them as fibroblasts. The main limitation, again, in making them into iPS cells has been, quite frankly, just the cost of making one cell line. Right.
You have the blood tonight, and then there's also a biomarker study that seems relevant also,
Yeah.
right? Where those patients are given blood and spinal fluid.
Right.
That,
So
maybe-
biomarkers are absolutely, so we have a plan designed right now to actually take patients to get all the things you mentioned, blood, spinal fluid, skin cells, and make iPS cells from every one of those patients. That way, I told you about personalized medicine. So if you had a bulbar onset ALS and you were a woman in your 50s and had a progression that took 5 years, we would be able to match up all those patients from a biomarker standpoint and say what's unique about those patients. Do those patients have abnormalities in their motor neurons, or maybe do those patients have abnormalities in their astrocytes? So those are really, good questions, and that's where we'd like to see this go.
Okay, we have time for one more question. Where's the mic?
With the iPS cells, you talked about using them in the dish to learn something about how patients will respond to drugs. I wonder if, in a sort of an extension to that, looking at simply how those cells would respond without drugs in order to make predictions on progression. Because it seems like with clinical trials, that baseline is one of the big issues.
So that's an excellent question. It's so excellent that we're actually doing that in our laboratory. No, so what I'll tell you is, I didn't show you a slide, but we're actually doing that same paradigm I just showed you. We're actually able to make these cells and to make motor neurons and follow them out for 100 days and to ask questions. We can actually sample the fluid in the dish and ask a question, how are those cells dying, how fast they're dying, do they send out the same number of axons or cells? So absolutely, that's all. And so those are questions we're very much interested in. We're actually able to stress the cells, so we're able to stress them using a variety of chemicals to say, does one cell type change from another? So absolutely. They only gave me, whatever it was, 45 minutes, but okay.
Great, thank you. We might have some time at the end of the session for questions, but I want to make sure our second topic is also addressed. So our next project, the federal government, we've been working on we ourselves, the association, for almost a decade, is our ALS Registry. And I want to share a little bit about the registry because one of the things that I really learned by working on this project is how many different definitions there are of the word registry. And for some people, it really is basically all about numbers. It's basically spreadsheet, that's what it is, boom. That's what we want to get out of. When we came together and worked with a lot of researchers throughout the country about the registry, we decided that we needed more than just collecting the numbers. Connecting the dots is important, but we also need a powerful research engine. And in fact, that's one of the reasons why whenever we do a priority, we always try to be collaborative with our other neurological diseases out there, especially. And it's one of the reasons why the MS Society didn't join our sort of plan, because there had been some work at ATSDR looking at ALS and MS and the instance, et cetera, because they felt they already had a powerful research engine. So I say that only in this session. We'll have more in our next session about how we introduce the bill, et cetera, is for a lot of people, there's a lot of differences and a lot of definitions that are different about the registry. So while the numbers are important, the powerful research engine that we wanted to create is also important. And one of the things that we've learned is that the folks at ATSDR, CDC, are very responsive. So I encourage you to listen to Dr. Kevin Horton, who is a project manager at the registry, and also share with him. I know some of you aren't shy, but with the folks that we've really begun a dialogue with CDC, they've been very responsive. And even on things that might seem not as important to a government bureaucrat like the password issue that we heard from you early on and we changed it. Those are all important to people with ALS and their families. So please listen and please share. Dr. Horton?
Good afternoon. Okay, let me see if I can figure this out. Okay, I got it. Okay, first of all, I'd like to thank ALSA for inviting us back. It's always a good opportunity to educate the stakeholders about what we're doing at ATSDR CDC. And so for the last 3-4 years, you've heard me up here year after year talking about the registry and what we're doing and how we're progressing. Well, I'm here to tell you today that next month we're going to have our first report with numbers, prevalence, demographics, the whole 9 yards, what we've been working on for the last 3 years. And so we're very excited. I'll go through some of what we're going to be presenting in the report and talk about some of the other things that we're going to be unveiling this year. So again, I think most of you know that the Agency for Toxic Substances and Disease Registry, ATSDR, we are a sister agency of the CDC, and we are the ones who have created and maintain the National ALS Registry. So again, I apologize. Some of you, a lot of you have probably seen these slides, but I know that there are some people out there that don't know a lot about the registry. Maybe you're new to ALS, and so I just want to make sure that everyone understands. So the ALS Registry Act was passed in 2008. Most of the credit, I'd say all the credit, goes to ALSA and many of you in the room for really getting this law passed. And what it does is this really kick-started the registry effort. It allowed CDC, ATSDR to come in and establish the registry. And, you know, let me say, and Steve said this, there are a number of different definitions for registries. That's true. It seems like there are 3 or 4 different ALS registries. But this one is the only population-based registry for the entire United States. So I just wanted to make that clear. If you look at the law or the bill language, it specifies 3 primary things that this registry should accomplish, looking at the incidence and prevalence of ALS. So in other words, how common is ALS around the U.S.? We have an idea, or we have ideas because of smaller scale epidemiologic studies, but these are not population-based efforts. They're small-scale studies, and the numbers are extrapolated out to the U.S. to give us estimates of how many cases we'd expect to see. So really, this is going to help us more accurately describe what is the incidence and prevalence of this disease. Who gets the disease? You know, how does it affect various populations, whether it's males, females, Caucasians, African Americans? We want to know who gets the disease. And I think one of the most important things, and I think most of you will agree, is why do people get this disease? You know, Jane said it earlier, you know, Lou Gehrig was diagnosed 75 years ago. Progress has been made, but there's still a lot that's unknown, and a lot of it has to do with the etiology of the disease. So we're hoping the registry will help us look at the risk factors for the disease and hopefully point scientists or researchers into the direction that we need to be looking at. And the registry itself, the online portion, was launched in October 2010. Okay, so let me just briefly go over this. So we take a 2-pronged approach for capturing or identifying ALS cases in the country. So remember, ALS, like many non-communicable diseases, it's not a reportable disease in the U.S., okay? So for that reason, ATSDR CDC, we had to come up with sound scientific methodology for trying to identify all the ALS cases in a country of 300 plus million people. So not an easy task, but we've come up with a couple different strategies that are really helping us identify cases of ALS around the country. The first approach on the left-hand side is an approach where we're using existing data sources, administrative data sources such as Medicare and Medicaid and data from the VA. And what we're doing is we are applying an algorithm, an electronic algorithm to these databases that will allow us to put people into different categories. Now, obviously, we're interested in those that meet our case definition or that are identified as a true ALS patient. Those people will go directly into the registry. And for those that are potential cases, that just means we don't have enough information one way or the other to classify them, yes, this is a case, or no, it's not a case. So for this approach, patients don't have to do anything. So the approach on the right-hand side, I think, is the one that most people in this room are familiar with, and this is the approach that launched in October of 2010, and this is the web portal approach, or the self-enrollment approach. So in a nutshell, you go to our website and you answer a series of validation questions, and depending on the way you answer, again, you're either classified as a true patient in the registry's eyes or not a patient. Those that are patients will have the opportunity to answer some brief risk factor survey questions about, what potentially could have led to the case of ALS. Things like drinking history, smoking history, occupational history, military history, things along those lines. And so it's really important for us to encourage patients with ALS, especially those newly diagnosed, to come and enroll in our registry, because it will help us not only figure out how common is this disease around the U.S., but really let's look at what are the risk factors? Are there common themes? And the more people who enroll, the more robust the data will be, and the better chances that we have in identifying a risk factor or risk factors for ALS. Again, just wanted to show you joining the registry is pretty quick, painless. Go to our website. It's fairly intuitive. We've gotten feedback from PALs about how we can make the registry better, especially the online portion, and so we've tried to take a lot of these suggestions and implement these suggestions. Once you enroll, you're able to, as I mentioned, go forward and take various survey modules. And this is just a snapshot of what it looks like. Once you complete the module, it'll tell you, it's completed, or if you have a module that's yet to be completed, it'll say something like in progress, or, you need to take this. So let's talk about the current risk factor modules. When we launched the registry in October of 2010, we launched with 7 risk factor modules. And these 7 are the ones listed in white, the first 7 here. And I'm happy to say that since then we've launched 5 more. 3 were launched last week. And again, we're trying, whenever we launch something like this, we try to go through ALSA, we try to go through MDA, Les Turner, the various ALS organizations, to let them know, to let their stakeholders know. Again, and you'll hear me say this a lot, this is a collaborative effort. We cannot do this alone. We can't do this with just ALSA. We have to have all hands on deck. And so happy to say that just since last week, hundreds and hundreds of people have already come and taken these surveys. So we really appreciate that. If you can help us spread the word to other PALs that have not enrolled or maybe don't know about these new survey modules, we would really appreciate it. Okay, so this is the question that's the burning question. And I want to explain, why we are where we're at right now. We get this question a lot, the heck's taking so long? And that's a valid question. But if you go back to that chart that I showed you, this is a two-pronged approach. Most of the cases that we get in the registry come from Medicare. Okay, so Medicare is a data source that we have to make the registry work. Our first data set next month, our first report, will be for calendar year 11 data. So we launched the registry in October 2010. Typically, the way that registries work are on a calendar year. They release a calendar year's worth of data. Cancer registries do this, birth defects registries do this, and so it's going to be calendar year 11 data that we will describe in this report. So we just received calendar year 11 Medicare data this past fall, which tells you that there's a lag time between the time we get the data and the time we're able to implement, put the data into the registry. There's usually a 2-year lag time. The good thing is the admin data has finally caught up with the portal data or the web self-enrollment data. So the good thing is we're on track now, and I was just told last week that Medicare, Medicaid data for calendar year 12 is already available. So we're in the process of getting that data, and as soon as we get this report out, we'll start on the calendar year 12 data. So all that's to say is we should be on track, and every year we'll be able to release a report along with a new data set. So how are we going to do this? So the first report right now is in clearance. As I mentioned, we anticipate it being released next month. We're going to publish it in the CDC publication. CDC has a publication called the Morbidity and Mortality Weekly Report. It's CDC's journal, if you will. And so we're going to release it in this journal, and we're doing this for a number of different reasons. If we tried to release it in a typical public health journal, we don't really have control over when we can get that data published. But with CDC being this is our journal, our goal is to have this released every year at a certain time. Hopefully every May we'll be able to release a new report. And so this is where we're at. As I mentioned, the first year is calendar year 11 data, and that will consist of the web portal data combined with the administrative data. And so we shake out all the duplicate people in there, because we know that we'll find people in Medicare, Medicaid, and VA, and then we'll also have people come, the same people come and enroll in the web portal, and that's fine. We can cross-reference on a matching ID and make sure we're only counting people one time. So I really just wanted to make that clear, that it's not that we're twiddling thumbs. We're having to wait for the administrative data to catch up with the portal data, and it finally has. So we're moving forward as quickly as we can. What I can tell you right now is the data from this first report is very consistent with the data that you've seen in the literature, especially the data from the long-standing ALS registries in Europe, and some of the smaller scale epi studies that have been conducted in the US. So I wanted to give you a little bit about some of the data that you'll be seeing. So the data that you're seeing now, this is from 2001 to 2009. And again, this is administrative data sets. So this is the Medicare and Medicaid data sets, and a couple of data sets from the Veterans Administration. And the point of this is to show that the algorithm is working, that we're able to identify thousands and thousands of ALS cases. This particular slide is not complete, and so that's why I have a little disclaimer down at the bottom. The Medicare and the VHA data in this slide are from 2001 to 2009, but you can see in the little footnote here, we only have Medicaid through 2007 and VBA through 2005. So I just want to make that clear, and the point is that it's critical that we have a very sound and scientific algorithm to help us identify ALS cases in these data sets. Again, this kind of reiterates what I said. A bulk or a majority of the cases that are going to be used to populate the registry come from Medicare. So again, very critical that we have this piece before we can enumerate anything. Medicaid, very few cases come to the registry through Medicaid. The VA, again, a lot smaller. And then there are people that come through in multiple sources. Again, this goes back to what I said a few slides ago. The data that we're seeing in our registry, very consistent with what is known about ALS. Again, I think it's critical for us to really describe the ALS experience in the U.S. You know, it's great that we can look at data from Europe and other places, but this is really describes the ALS experience in the U.S. Again, this slide just shows you that, you know, this is a disease that typically affects those that are in the 60 to 79 age category. Obviously, we know that there are cases that are much younger, and it seems like every time I come to this conference or another conference, I'm hearing of more and more cases that are younger and younger. The point of this is just to show you kind of the age distribution or breakout. And we'll break the data out like this for our first report as well. Again, no shocker here. This seems to be a disease that affects primarily Caucasians, although we know that it does affect other groups. But predominantly, this is a disease that affects whites. Again, it's more heavily affecting males than females. Again, this data fits with what's in the literature. So this slide I wanted to show because I think it drives a point home. So this slide here looks at the percentage completion rate of our surveys. And so if you can see here, hopefully you can see the little red lines, this is where we were last year. So this is where we were last year in January. But since we have really hammered home the point that, hey, we really need PALS to not only enroll, but to take these surveys, we've increased the percentage of people who are taking these surveys. And this is really good. So approximately about a 5% increase across the board. Again, this is not because of CDC, ATSDR. This is because of chapters. This is because of MDA groups. You're out there encouraging PALS to enroll. And this is how we get people to enroll and take the surveys. Overall, the response rate here, you might think, wow, that's not too great. But in the grand scheme of things for federal surveys, this is actually really good. Could it be better? Of course. But I think it speaks volumes that this is a group or a population that really wants to find out what's going on. And so people are stepping forward to not only enroll, but to take surveys. So again, if you can help us encourage people with the disease to step up and enroll, it really strengthens the science. This is the percent of those completing the last survey, or I'm sorry, the disease progression survey. So this is a little smaller percentage. Again, This is a survey that can be taken multiple times. So of all the 12 surveys we have up there, 11 of those can be taken one time. This disease progression survey is one that we want folks to take multiple times a year. And we've since increased it from twice a year, I believe, up to 3 times a year. And so when it's time for you to take the survey, you will get an auto-generated email saying it's time to take the survey. So again, really encouraging people to really take the survey. You know, it's easy, it's painless. Again, and I was talking to a patient this morning. If it were up to us at CDC, ATSDR, if it were up to me and I ruled the universe, we'd have a million questions to ask. And we've heard it from folks, hey, this is good, but you haven't asked about X, Y, Z. So I'm going to show you some new surveys that we're going to be releasing later on in the year. So we are trying to listen to folks and to researchers to figure out what are the other questions that we should be asking about. So I wanted to talk about a couple things that I think are really important for the registry. As Steve mentioned, you know, it is important to enumerate, you know, the number of cases in the U.S. That gives us a baseline of, you know, how many cases do we expect, how many cases are we seeing. But the registry can be used for a lot more different things than that. And I'm just going to go through some of these things to talk about what we're doing and how we're using some of these initiatives to strengthen the registry. The first thing that we're doing right now is, or we have been doing since 2012, is we have opened up the registry to researchers to use as a recruitment tool for new clinical trials or epi studies that they are about to bring online. We know that PALs want to take part in research studies, whether it's clinical trial or study. We know that they want to take part in this. And so we thought, you know what, this is the largest database in the U.S. Let's open it up right now. Before we even release our first data set, let's open it up, make it available to researchers to use. And so this is the way it's working is, and I've got a little schematic that I'm going to go through, but we're essentially linking PALs with researchers. And right now, PALs who want to take part in this can check a little box during enrollment saying, yes, I want to be notified about new studies that come online. And the good thing is approximately 96% of all enrollees in the registry have checked that little box, which tells you right off the bat, people want to take part. They want to, you know, they want to find out what has caused their disease. But in order for this thing to work, we have to have PALs sign up for it. And we have to have researchers submit proposals. Now, again, it's not mandatory that you know, the patient takes part in this. It's totally up to you, and want to make that clear. So how does this work? So when patients enroll in the registry, as I mentioned, there's a little checkbox that you check saying, yes, I give ATSDR CDC permission to contact me about new studies or research opportunities. Once they do that, once PALs do that, that's it. There's nothing else you have to do. On the other end, researchers come in, they come to our website, they fill out a pre-designed questionnaire that essentially tells us what is your study about, do you have IRB approval. We really want to make sure it's a valid study and that, you know, it's on the up and up. We have an internal/external review committee that reviews these proposals, and if the proposals are approved, depending on what the study criteria are from the researchers, we're able to carve up the data, so to speak, to reach that subset of patients. So let's say, for example, Nick is interested in doing a study on all African American PALs who have been in the military. We can slice the registry up, focus just on that subset of patients. We can then push out the study protocol, the PI contact, all the information about the study, and then it's up to the PALs to reach back and contact the researcher if he or she wants to take part in this study. So you can look at it as ATSDR, CDC, we're kind of the middlemen. We're not funding these studies, we're not, we're just linking PALs with researchers. And so right now, there have been 8 institutions that have taken us up on this. And some of these are people that are movers and shakers in the ALS world. We're about to push out another study on Friday. And so, some of these studies are clinical trials, others are epi-type studies. But to date, we've pushed out over 13,000 email notifications to patients in our registry, letting them know that they're eligible based on the study criteria, they're eligible for participation in these studies. And again, just want to emphasize that you can go to clinicaltrials.gov, and I think probably many of you, if not most of you in the room, have been to this. It's not the easiest website to navigate. And what we're trying to do here is to really bring the study to you, to your inbox. And that way, you don't necessarily have to stay on clinical trials or frequent clinicaltrials.gov. And this is really, in my opinion, this is where the rubber meets the road here. These researchers are doing some really good work, and we are not only trying to market the registry to patients, but we have to let our researchers and neurologists out there know that we have this capability, and the registry is there to use to help them in their studies. So we're very excited about this aspect. We're going to bring more risk factor surveys online later this year. The first 3 have been told will come out by or before August, and then the other 2 will come out in the fall. Again, this is not a complete list. We may have other surveys come online in the future. If a researcher out there says, hey, I think you should look at this, or PALs come to us and say, what about this? It's something that we can think about bringing on new modules. And again, this is really there to help us learn more about what are the risk factors for this disease. We are in the 3rd year of a 4-year biorepository pilot project. And again, I know that, or we know that there are ALS biorepositories out there. And so we're not trying to duplicate what other people have done, but we are trying to figure out, does it make sense to fold a biorepository into the National ALS Registry? And obviously I'm a little biased, but I think it makes sense because if you think about it, if we collect a biological specimen, whether it's hair, nails, blood, there are not too many biorepositories out there that actually have the epi data to go along with these samples. So the epi data that I mean are the military history, the occupational history, residential history, that type of data, when paired with a biological specimen, very powerful data there. So this is where this biorepository would differ from other biorepositories. These are the specimen types that we're collecting right now. So we're doing this in 2 different phases. We're doing what we call an in-home collection and then a post-mortem collection. So you can see in the first graph up here on the left-hand side, we're collecting blood, urine, nails, hair, saliva when blood can't be, sorry, I'm getting ahead of myself here. We're collecting these things, and we go out to homes to collect these. So we don't make PALs go to a referral center. Again, we're trying to look at specifically the logistics of how we would stand up a biorepository. And the easier we can make it for PALS, I think in my mind, the better. And so the same with postmortem. You can see here some of the biospecimens that we've been collecting. And then down at the bottom, just wanted to give you some numbers. PALs have been very receptive. We've had a lot of PALs call us up saying, hey, I want to contribute my whatever blood, and we appreciate that. So far, you know, over 400 samples collected, in-home samples, and then we've gotten 3 postmortem specimens. So I think this speaks volumes about PALs and their caregivers. People want to learn about this disease. And, you know, what a selfless thing to do. You're giving up, you know, it just amazes me what this group does and the patients and what they go through. So again, this study will conclude next year. We'll decide at that time, should we incorporate a biorepository into the National ALS Registry. And really, we're going to try to let the science dictate what we should do. State and metro-based surveillance. This is another exciting component that does a number of different things. The first thing it's going to do, the biggest thing it's going to do, is really help us test the completeness of the registry. So what do I mean by that? Well, there are 3 states and 8 metro areas have taken part in this, in these surveillance efforts. And so what we've done is taken an active case surveillance approach. And so we've gone out and canvassed neurologists in these areas and actually have gone and abstracted all the ALS cases that these neurologists in these areas have seen. And so what we're going to do is take that data, compare it with the data we have in the National ALS Registry to help us test, you know, are the national data complete? Are we seeing more cases from these areas than we are compared to what we have in the National Registry? If so, then we need to come up with, tighten our algorithm, come up with a way that we can make sure that we're getting as many cases as we can. So the data that we collected was for 2009 to 2011. So what's the status of this? So the surveillance activities have concluded. For those of you who are interested, you can go to our website, and we have an executive summary for the state that's on our web portal. As I mentioned, we're going to begin comparing the state and metro data with the national data this summer. Again, our first priority right now is to get this first report out the door. And once we do that, then we'll be able to look at the comparison aspect. But also, what this is doing is this state and metro effort is really helping us look at incidence and prevalence in smaller geographic areas. And so we've got a number of different publications that are in progress right now, some of which have already been submitted to journals. Again, these will be things like state and metro incidence and prevalence. I mentioned earlier who gets the disease. So we'll really look at racial and ethnic differences of ALS. Case verification, you know, are the cases that are seen, are they actual ALS cases? And then looking at death certificate and using death certificates as a way to enhance the registry. Death certificates are good, but they're notoriously unreliable. So you can't necessarily read on a death certificate that someone's died of ALS and that's an actual case. And there's been a number of studies in the literature to show that. And so we're really looking at how can death certificate data help the registry. And I think really the conclusion from this surveillance system is it can be done, but if you do it on a state by state or case or metro by metro area, very expensive. To entice neurologists to take part in this, we had to pay neurologists on a case-by-case basis. And so not every neurologist wanted to participate. So, you know, even though you're paying people, it doesn't mean that they're going to participate. So not only is it expensive, but it's very labor-intensive. And so I think this really drives home the fact that a national approach is a more economical approach, and we're getting actually more bang for our buck. And again, all of this will come out as we write it up, but this is very exciting for us. And again, this is going to be a big year, not only for the report, but for all of these articles and reports that are going to come out in the state and metro. And again, this is just, I just grabbed, this is New Jersey data. New Jersey was one of the states that took part. If you look at the crude incidence rates, pretty much what's in line with the literature, about 2 per 100,000. Prevalence, about 4 to 5 per 100,000. So these are the type of data that we're going to be able to publish and really build the literature and build the evidence for what is the ALS experience in the U.S. And keep in mind, we have 3 states and 8 metro areas, and all will be providing data, such as what you see up here. Here we go. Okay, research funding. Research is critical for understanding ALS. Again, the registry is not all about enumerating or trying to, pin a number on how many cases are out there, but really funding research will help not only the ALS community at large, but it will help ALS or ATSDR prioritize topics for future risk factor surveys. So for example, if, cyanobacteria is something that's been mentioned in the literature as being potentially associated with ALS. This is something that we can explore by putting together a risk factor module to help us find out more about, cyanobacteria, caffeine consumption, statins, things that haven't really fully been explored. And so things that we learn through research will really help us in the registry, help us refute or validate or corroborate or whatever. So we have funded quite a few research studies. Again, these are some more movers and shakers, and all of this information is on our website, and we're trying to keep an updated list, so every time we fund a new study or every time someone uses the research tool, recruitment tool, to let people know, hey, we're getting a lot of use out of the registry. So here's some of the research that we're funding. We're going to bring a couple more studies online this summer. People have asked us, how do we spend the money that we get? And I think that's a valid question. Right now, I will tell you that a lot of the money that we spend goes for education and outreach. So again, you heard me say that we cannot do this alone. And so a lot of the education and outreach comes from you guys, comes from the ALSA chapters, comes from the MDA chapters. And you guys are the ones on the front line. You interact with patients on a day-to-day basis. We don't. We don't see them. I see them when I come to conferences like this or other conferences. So in order for this registry to really work, people have to know about it. And so this is what some of the money goes for, is education and outreach. I mentioned research. We have to fund research. So those two are kind of the bulk of what money's being spent on. This is for FY13, fiscal year 13. FY14 is largely the same. FY15, which will start October 1, part of this new funding that we get could potentially go for a full-blown biorepository, but we don't know yet. We have to wait and see what the feasibility study says. So this is just a snapshot of how money are being spent with the registry. Wanted to mention a couple extra things, or a couple of final things. It's great that we interact with PALS, but like I said earlier, we have to interact with so many more people, namely neurologists, researchers, And so we put together back in 2011, I think, continuing education module. And this is really to educate healthcare providers who see ALS patients, to really educate them on ALS, the registry, and healthcare professionals are always looking for continuing education credits. I know I am. And so this is a quick way to get free education credits. And to date, we've had about 800 healthcare professionals come through and take these continuing education module to get credit. So again, if you know any healthcare professionals out there, nurses, physicians, whomever, that are interested in getting continuing education credits, this is a good way to get it and learn more about ALS and the registry. Just wanted to mention a couple of other things that we got. We just recently released last week on Google Play the Android ALS Service Locator app. And really, this is meant for those that are new to ALS or maybe newly diagnosed or just want to find out more about what's in the area. And so you can go to either the App Store, the iTunes App Store, or Google Play and download this. Put in your zip code, and up pops the 5 closest ALS clinics, or depending on how you choose it, the ALSA chapters or MDA chapters. So again, trying to make a useful tool for patients to find support in their area. About, I don't know, what does it say there? About 455 people have downloaded these apps. And again, if it helps, great. Just trying to make it useful for patients. As I mentioned, later this year, we're going to start publishing a lot. You can find a lot of this, all of this on our website, so I would encourage you to go to our website. We have links to reports. Many of you have already looked at our reports for our annual surveillance meetings. So whenever we get a new journal article published, we'll put it up there. Again, this is where it's at right here. It's not about me. It's not about you. It's about everyone in here talking about ALS, promoting ALS, what a devastating disease it is, and collectively coming together to address ALS. And can't do it without you. Can't do it without ALSA. Can't do it without the likes of Nick and other people. So we all have to pull together here, and the more people that can come in and help us, especially take part in the registry, the self-enrollment part, the better the data are going to be. I believe that's, oh, so take-home message. Sorry, last one. First report, very soon. Many journal articles are going to be coming out. Survey completion, it's increasing. Can it be better? Yes. But again, we're pleased with where we're at now, but it can always be better. More risk factor surveys coming online. Talked about the biorepository collecting hundreds of samples, state and metro surveillance, really helping us find out the completeness of the registry, research funding. And again, I'll say it one more time, can't do it without you guys, and everyone in here is critical to the success of the registry. And with that, I will take questions.
Okay, this mic's start over on this side of the room. Before the mic gets over there, quick question, Kevin. There's an open-ended question. Can you share a little bit about that? Because I know many times when we meet with patients, they all have their own idea or theory on perhaps how they were.
So we know that every ALS, every pals out there, you have a story to tell. And so for that reason, we created these open-ended questions, and it's really 2 questions. You know, what do you think contributes to ALS? And then what do you think contributes to your case of ALS? And again, there may be things that you put down on there that we haven't thought of or we haven't heard of. And so this is a good opportunity for pals to, you know, tell us what you think. And so this is the reason that we created these 2 open-ended questions.
Okay, great, thanks. How about over here?
Hi. We get a lot of questions in our support groups, and I know that this has been suggested before, but I'm going to reiterate it again. It'd be really cool if those of us who interact with patients could kind of see what's out there. I know my dad died right when the registry came on board. I have not been through it. I can't get into it. And therefore, when I have a patient that asks for help, you know, I kind of look like an idiot. Is there something out there that we can see? Is there a download of the type of questions that we could at least be somewhat intelligent with?
Right.
And then secondly, kind of going into that, some of my pals have actually said to me, what's in it for me? And I don't think, not in remuneration necessarily, but what access will they have to some of the data now that all this is coming out in June, which kicks into some of my legislators have asked to see what's being published.
Right.
So how easy it is for rank and file to get to that.
Right. So I guess I'll answer your first question. The data, I mean, we're going to make it available in a report. We're going to also put fact sheets out, kind of summarizing our findings. We're going to have a data set available. It's going to be a de-identified data set that researchers can use to help describe, you know, what's going on with the registry and the numbers that we're finding. So we'll be, you know, all this will be coming out. What's in it for them? That's a hard question to answer. I would say what's in it for maybe the next generation or those that are going to be diagnosed with ALS in the future. Maybe the registry will lead to, I don't know, some finding or, you know, risk factor that we didn't know about. So that's kind of a difficult. And clinical trials, right? So, you know, if patients are interested in clinical trials, they can sign up for, you know, the mechanism that I mentioned, and be notified about, you know, what they can take part in. Remind me what the other question was. Oh, right. So right now, what I would say is the more times you can help a patient enroll, the more familiar you will become with the registry and with the questions. We've been asked to provide the laundry list of questions, and we've thought about doing that, but after discussing it, we think it's best to hold back, and the reason is we don't want to introduce bias into the way people answer questions. So, for example, if you know that there is a risk factor module out there on military history, you might think, well, you know what, I'm not part of that. I'm not going to bother answering that question. And so we would rather people go through it themselves and answer the questions and have you sit down side by side as a person in the chapter or even a caregiver, because the more times you do it, I think the better. I know that's not the best answer, but that's kind of what's been decided in terms of releasing.
Mike?
Hi, how you doing? My name is Wes Rose. I'm from the Greater Philadelphia chapter. Thanks for the update. I have a quick question about the pie chart, the funding pie chart. Can you go back to that? Thank you. I think all of us value the need for education and outreach for sure. Now that the registry is established and a lot of the ALS community is aware of what's going on, is there any kind of plan of shifting the allocation towards, say, research from? Because I think an allocation of over 50% to education and outreach is great, but I think we would like to see more research.
I absolutely agree. But it's kind of a slippery slope. You know, we don't want to necessarily take away from education and outreach because people need to know that the registry is in existence. So, you know, how do we do that? I'm there with you in spirit. I'd rather, you know, have a majority of this be researched. But I think the more chapters and MDA offices, the more they come to know about the registry and what the registry is all about, the more it becomes probably an everyday part of their job. You know, it becomes part of them to talk about the registry and the importance of the registry. And so maybe we could scale back some of the funding and put more in research. I wouldn't say that there's no, I mean, anything's possible. But again, we can have the best registry in the world, but if people don't know about it, especially newly diagnosed patients, we're kind of. Right. I guess I would let Alsa handle that question, but I totally agree, Wes. I'm there with you.
Where's the mic?
Is this on? How many people total are in the registry right now?
So this will come out next month, and
Ballpark?
this will come out next month. The information will come out in the MMWR. When I went to the editor and said, I'm going to ALSA to talk about this, we need to let them know it's embargoed. And I understand that's not a satisfactory answer.
No, it's not. We've been waiting
back
for a long time, and the lack of information, the lack of updates.
So
I have a follow-up question.
Okay.
Has the ALS Association and MDA and patients like me, have they provided their lists of PALs to the registry?
No. I mean,
Why not?
What they do, because it changes every day, and we work with them every day, and they know who are newly diagnosed. They know who has registered, who is enrolled. We don't, okay?
Yeah.
And so this is where the education and outreach comes in with ALSA and MDA,
So you're paying them to market the registry, but you don't have their lists? I don't understand that. You're paying clinicians for their lists. ALSA probably has the largest list of people with ALS in the country. Maybe somebody from ALSA can speak to that. Why doesn't your group have that list?
Well, I mean, each person has to enroll itself. So we have reached out to all the various chapters to enroll their folks. So there is no list.
But you're getting data from other sources where enrollment is not necessary, right?
Okay. There's a couple things here. One, the scientific part of this doesn't permit us to do that. Patients themselves have to enroll directly. We can't simply share a list. What we do is we go out to the communities across the country, to our chapters, everywhere across the country, to try to encourage people to enroll, because the science requires that individuals enroll. We can't simply provide a list. We would love to, but that's not the way the science works, unfortunately. So what we try to do is really, really promote this among patients, among families, caregivers, everybody who comes in contact in some way with a person with ALS, because we need to drive enrollment. The best way for this to succeed is that more people enroll. We would love to provide a list, but scientifically that's just not the way it works. But I certainly understand the science.
And also, let me just say something. We have to be careful with the IRB about coming across as being coercive. Not everybody out here wants to take part in the registry. So, while I want you to take part in, if you don't want to, I can't make you do that, and we can't, go up with a list and say, hey, according to this, you haven't enrolled. That's a big IRB no-no.
Yeah, I mean, there are informed consent issues. There's also privacy issues where we don't have the permission to share that information. I mean, that's another issue. Sure.
You don't have to put those people in the registry, but then at least you would know of the patient population what percentage you have captured in the registry. I don't understand why you can buy the information from the ALS Association. If you're a marketer, you can rent a list from the ALS Association. Yes, you can.
Not patients. Not patients. You cannot.
Okay.
Cannot. And in fact, interesting, when we started this registry project, we started in one state, Massachusetts. And in that state, we actually had it mandatory that, like some of the cancer registries, that it had to be reported by the health professionals. And we got a lot of angry patients saying, you're going to report this and I haven't told my family, I haven't told my work. So while we applaud you for wanting to share everything, there are many patients that don't want to share, and we have to respect those patients.
Yeah, okay.
There's a question back here. We have time for one more question.
Yes, my name is Sonal Shah. I'm from New Jersey, belong to Greater New York chapter. How do I find out a valid data how many patients are in New Jersey? Now, half of New Jersey belong to Greater New York, and the southern part belongs to Philadelphia chapter. Does it matter? I often hear from different sources there are over 500 patients in New Jersey, and certain counties have clustered. How do I know what is being done with that data? Why certain areas are clustered areas for ALS patients? My town has half a dozen patients right now with ALS.
Right. So, well, New Jersey was one of the state that we did the active surveillance, and so we have very good numbers on New Jersey in terms of the number of cases. But I think what you're speaking to, the registry, what the registry will do will give us baseline data about how common is ALS in a given area. And we could potentially use it for cluster type questions. You know, is this appear to be a higher number of patients than we'd expect to see that lives in this area? Right now, it's hard to answer because we don't have any baseline data. But gathering data through the registry will allow us to examine those types of questions. But I would say specifically for New Jersey, you'll have that information this year. And the New Jersey data that I showed you is on the New Jersey website. I just pulled it straight from the New Jersey website, the New Jersey State Health Department website, or you can go to the ALS Registry, our website, and get to it that way. And it'll tell you incidence or prevalence, case counts, things that you were talking about.
Okay, well, thank you. We're about 15 minutes over. Kevin is here the whole conference, so be happy to talk to you right after this in the break or sometime on Thursday or Friday. He'll actually be in our room on Thursday, our hospitality room too. So thank you.
Thank you. We are too, yeah. Our goal is to have it